Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Chiasmata explained. Formation, structure, and role in meiosis. Table of contents. Introduction to chiasmata. Formation of chiasmata. Structure of chiasmata. Measurement of chiasmata. Significance of chiasmata. Introduction to chiasmata. Chiasmata, singular chiasma, is derived from the Greek word meaning X-shaped cross. It represents the point of contact where two non-sister chromatids of homologous chromosomes are physically linked together. This contact occurs specifically during the Pachytene stage of Prophase in Meiosis Y. The chiasmata maintains a connection between homologous chromatids until anaphase O's of Meiosis Y, ensuring proper chromosome segregation. The phenomenon of chiasmata was first discovered and described in 1909 by Frans Alfons Janssens, a Belgian cytologist. Janssens introduced the term chiasmatopie to describe this crossing over behavior observed during meiosis. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Formation of chiasmata. During the prophase of meiosis I, homologous chromosomes undergo pairing and recombination. In this phase, crossing over facilitates regular exchanges between homologous chromosomes. Prophase I is subdivided into 5 distinct phases: leptotene, zygotene, pachytene, diplotene, diakinesis. Chiasmata form at specific sites where programmed DNA breaks, created by a specialized enzyme called SPO11, follow the complete recombination pathway to generate crossovers. The precise mechanism of how crossing over translates into the physical formation of chiasmata, which represent exchanges of DNA sequence data, is still not fully understood. The centromeres of sister chromatids are joined to form a single kinetochore, which attaches to microtubules during orientation of homologous chromosomes on the metaphase plate in meiosis I. Homologous chromosomes move very close together and form a structure called the synaptonomal complex just before the onset of the Pachytene stage. Formation of the synaptonomal complex initiates chiasma formation and is essential for crossing over and recombination to occur. The synaptonomal complex is a protein-based lattice that forms between homologous chromosomes and begins at specific initiation sites, eventually spreading along the entire chromosome length. This tight pairing of homologous chromosomes is known as synapsis. Crossing over, the exchange of chromosomal segments between non-sister chromatids of homologous chromosomes, is facilitated by the presence of the synaptonemal complex. Homologous chromosomes remain connected until anaphase of meiosis I due to cohesion between chromosome arms distal to the chiasmata, which resists the pulling force exerted by spindle fibers. At anaphase I, this distal cohesion is released, allowing chiasmata to separate, and the sister chromatids, with at least one crossover exchange, move together toward the same spindle pole. Consequently, the 2 daughter cells produced in meiosis I are haploid, each containing chromosomes with 2 sister chromatids, drawn randomly from the 2 parents. The number of chiasmata varies between species and depends on the length of the chromosome. For accurate and effective homologous chromosome separation during meiosis 1, there must be at least one chiasma per chromosome, though the number can reach up to 25 chiasmata per chromosome. Structure of chiasmata. The ultrastructure of chiasmata is still unknown, but it is believed that each chiasma consists of two unaltered sister chromatid arms, along with two recombinant arms that contain spliced DNA molecules and associated protein structures. The distal sister chromatid arms, which remain cohesive between the chiasma and the telomeres, help stabilize the DNA complex on the chromosome. A single chiasma is sufficient to link homologous chromosomes together during meiosis 1. In most species, the total number of chiasmata across both males and females is significantly greater than the total number of chromosomes. Humans have 39 such chromosome arms on their 23 pairs of homologous chromosomes, excluding the 5 acrocentric short arms, which generally do not undergo crossovers. Typically, only one chiasma is produced per arm in most cases. In human males, the number of chiasmata usually ranges from 46 to 53. In human females, a single chiasma can maintain stable homologous pairing for over 40 years, and it is released at the appropriate time as the oocyte matures into an egg. Measurement of chiasmata. The distances between neighboring chiasmata can be quantified in cytological units, such as microns, particularly in animal species, with cytologically advantageous meiotic chromosomes. It is possible to create a frequency distribution of these interchiasmata distances to analyze their spacing patterns. Unlike the typical exponential distribution seen in linkage distance distributions, the distribution of interchiasmata distances is often found to be modal, indicating the presence of a most common spacing distance rather than a random exponential spread. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Significance of chiasmata. During meiosis 1, chiasmata are essential for ensuring that homologous chromosomes attach to opposing spindle poles and properly segregate to opposite ends of the cell. At each chiasma, a chromosomal crossover occurs, involving the exchange of genetic material between the chromatids of homologous chromosomes. The absence of chiasmata during meiosis often leads to improper chromosomal segregation, which can result in aneuploidy, an abnormal number of chromosomes in the daughter cells. The presence of chiasmata marks the physical locations where genetic exchanges, crossovers, have been successfully completed. Individuals with mutations in the cohesion subunit gene are typically infertile due to a lack of chiasmata, which prevents the completion of meiosis. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!